Hey, Hammy here, back with video number 2, in which we're going to cover section 9.3 over the steps of mitosis and cytokinesis. Remember that the names mitosis and cytokinesis are not synonymous, that mitosis is referring to the division of the nucleus of the genetic material or DNA, whereas cytokinesis is the division of the cytoplasm, the cell actually splitting into two. Okay, during mitosis, sister chromatids are separated and distributed to the daughter cells. Okay, so we have mother cell is going to divide into two genetically identical daughter cells. Before mitosis begins, the chromosome condenses into distinctly visible chromosomes. We can actually stain them and see them with a light microscope. And each species of organism has its own characteristic number of chromosomes. On this chart that you can see that, and it's not, down here's Homo sapiens, here's humans right here. So if you look, we have 46 chromosomes, okay? Whereas a goldfish has 94, a fruit fly has 8. You can't really say, oh, the more chromosomes you have, the more complex or higher organism you are. It's not really the case. Where a garden pea has 14, but then a southern adder's tongue fern, okay, here's a plant that's got 1,320 chromosomes. So, and a simple little yeast cell with 32, where we have 46, which is pretty close. So you can't say that, oh, more or less chromosomes make you more or less complex. What is important in mitosis is that this chromosome number is maintained upon divisions. Okay, so you can't just split in half, because then you'd have half as many chromosomes. So in the S phase of interphase, we double the chromosomes, the amount of DNA. Each DNA makes a copy of itself. Okay, so you have 46 chromosomes, but each one now has identical halves, sister chromatids. And so we're going to pull them apart into separate cells so that when you're done, each daughter cell has those 46 strands of DNA. They have all the copy of the genetics. Okay, when we say that humans have 46 chromosomes, okay, this would be our diploid number, or 2n, whereas n represents haploid number. Okay, so for example, of your 46 chromosomes, okay, 23 of them came from your mother, and 23 of them came from your father. Okay, so you have 23 from each. Those 23 types of chromosomes are represented twice, okay, because the sperm brings 23 from dad, the egg has 23 from mom. So you combine them together, and you've got one, like chromosome number 1, you have two, you call it pair 1, because you have 2 chromosome 1s, 2 chromosome 2s, 2 chromosome 3s. And we can actually, when we stain these, and in Bio 1, we look at that, what we call a karyotype, right? Where it shows from biggest pair 1 of the bigger, longer chromosomes all the way down to pair 22, and then your 23rd pair is your sex chromosomes, either XX or XY. Okay, so one set's paternal, one set's maternal. Okay, during interphase, preparation for cell division begins, and some of that includes this making of a centrosome. Okay, at the end of the S phase, each chromosome is duplicated. You have 2 identical double helical DNA structures. Okay, and they're attached, okay, kind of in the middle by a centromere right here. They're attached in the middle by a centromere, and each one of these is called a sister chromatid. Okay, sister chromatid, they're identical copies. Okay, during mitosis, these centromeres that hold these 2 together will split. That way, the 2 sister chromatids will become a daughter chromosome as they're pulled to each of in the opposite direction. They'll be going to opposite nuclei, so they'll each go to one of the daughter cells that is forming. Again, this slide just showing you kind of a microscope picture of that condensed chromosome with the centromere region here in the center. These are two sister chromatids. So in other words, they are exact copies of DNA of each other. So one chromatid, sister chromatids. There's also this region, this protein right here called a kinetochore. That's where the spindle fibers, in a second here, you'll see in a minute here, where they reach out and they attach one from each side. And they're as the centromere splits, they're what's going to pull them to opposite directions. That kinetochore is that attachment place for the spindle fibers. Okay, we mentioned that centrosome. It's usually close to or just outside of the nucleus, where you have the MTOC, the microtubule organizing center. The microtubule organizing center is what, sort of lines up the spindle fibers. Okay, and it also makes these two barrel-shaped things called centrioles. And they're oriented at right angles to each other, kind of like this. And they will move. There will be two of them on the opposite ends of the cell, and they're where the spindle fibers will anchor in and reach across the cell, and then you'll have your chromosomes down the center, something like this. So the centrosome, or the microtubule organizing center, is what's going to be responsible for making the centrioles in animal cells and organizing this mitotic spindle that you see over here that are used to pull the chromosomes apart. And each fiber is a microtubule. Okay, microtubule. Now, the stages of the whole cell cycle we refer to as ITMAT, okay, where interphase, okay, this is interphase here, and then this is mitosis here, where PMAT standing for prophase, metaphase, anaphase, telophase. Okay, so we're going to start right here at the beginning with prophase. This is where nuclear division or mitosis is about to occur. The chromatin condenses. It's distinguishable with a light microscope. We can stain them. We can see them. And they're two sister chromatids held together by a centromere. The nucleoli disappear. The nuclear envelope begins to fragment. And you get those spindle fibers starting to reach across the cell. Okay, and the two centrosomes move away from each other so that they will form centrioles kind of at the opposite ends of the cell, so they begin to migrate to the ends. And in animals, they kind of anchor in. You'll get the star ray. And when we look at the whitefish blastula cells in class, you'll see this little star-shaped array called an aster that kind of anchors them in on each pole. And after each one of these steps, I kind of have a slide here that shows how that cell looks. And you'll see that always on the top is going to be the animal cell, and on the bottom is showing a typical like plant root or plant onion root tip, which would be a plant cell here on the bottom. Also more of a rectangular shape up here, more of a round shape. You'll notice that in prophase, centrosomes have duplicated, okay, here and here. They're moving to the opposite ends of the cell. The chromatin in an interphase cell, you'll see just kind of a nice lightly shaded nucleus. In prophase, you can tell prophase apart. It looks like a normal cell here happening here. But in the nucleus, the DNA looks clumpier. Okay, same thing down here. Early prophase, you can see that it's starting to look kind of spotty, kind of clumpy. Again, next, just showing a little bit later in prophase, you see that the nucleus has completely broken down. Okay. And you get like, this is a good example down here where there's no nucleus, no nucleolus, and they just like these dark, yarny-like looking chromosomes in here. Up here, this is late prophase. This is, I would actually say, the start of metaphase here. They're starting to line up. But you see your two asters over here, kind of this, the centrosome with this array of microtubules kind of anchoring them in, and then all these chromosomes kind of clumped up right here in the center, but no more nuclear membrane. Okay, next step, pro-metaphase. Sometimes we split metaphase. We add pro like right before metaphase. This is preparation for sister chromatid separation. The centromere of each chromosome develops that kinetic core, which I showed you on that previous slide. Okay, so when you have your two sister chromatids with the centromere in the center, okay, that kinetic core is a little protein on either side where the spindle fiber can reach out and attach to. One is attached to each sister chromatid. They physically connect sister chromatids with specialized microtubules called kinetic cores. And again, these are used to pull them to opposite edges of the cell. So this slide here showing pro-metaphase, where you get kind of this clumping and you start to see these spindle fibers starting to form up here. Okay, down here, you can see the difference now here from prophase. The nuclear membrane is completely gone, and you start to get kind of this lining up. These spindle fibers are starting to attach and organize those chromosomes right in the middle. Metaphase is one of the easiest ones to tell because there's a metaphase plate. It's a plane right through the center cell, right where the mother cell will be divided. The polar spindle fibers run from all the way from one side to the other, and the ones that are attached to the chromosomes will all be lined up. And again, remember the checkpoints we talked about in the last video. This is the M checkpoint. It will not let anaphase start until all of them are lined up, all the chromosomes are lined up, and the spindle fibers are attached to all the kinetic cores properly, because you don't want to start stretching and pulling without one being attached. It'll be left behind. You'll get too many chromosomes in one daughter cell and not enough in another daughter cell. It can really mess that up. So this is where one of those checkpoints happens between metaphase and anaphase. In anaphase, the centromere dissolves, releasing the sister chromatids, and the sister chromatids begin to break apart. Okay, now they're called daughter chromosomes, and they start being pulled opposite directions along the kinetic core fibers, along those spindle fibers. And the long polar spindle fibers will actually take the cell and kind of push on the ends, will start to stretch that cell apart and prepare it for division. Okay, again, slides kind of showing these in the animal and the plant cell. And again, easily recognizable because when we look at these in class under the microscope, because your chromosomes will have this thin line down the middle where all those sister chromatids are all lined up down the middle of the cell so that the spindle fibers can attach to the kinetic cores. So in the plant and the animal cell, you'll see this nice metaphase plate down the middle. In anaphase is another kind of easy one to recognize because now there is a gap or a space as they begin to be pulled apart. So anaphase is this pulling apart, the splitting of the centromere, and the sister chromosomes, now daughter chromosomes, heading different directions. So in both of these, you can see that little gap here in the whitefish blastula, the animal cell, and also in the onion root tip down here. The last phase, telophase. Telophase, the spindle disappears. The new nucleus envelope begins to form. So you start to get a nuclear envelope form around those daughter chromosomes that got pulled to either end. There's two clusters of daughter chromosomes. And again, you should have each type of chromosome. You should have 46 of each. Clusters are incipient daughter nuclei. The envelope forms around them. Okay, and they each have one type. And then division of the cytoplasm then begins with cytokinesis. Okay, chromosomes become diffused back into chromatin. Once that nuclear membrane forms, they can start to unwind into euchromatin and into more of a usable type and become functionally active again in the cell. Okay, again, looking at telophase underneath the microscope, you can see that there is here the cleavage furrow beginning to form. These are pulled completely in opposite directions. They're in opposite ends of the plant, of the animal, and you'll start to get a nuclear envelope starting to form around each one again. And the cell is also kind of being stretched and pulled apart as well. The chromosomes will begin to turn back into chromatin and not be so coiled and condensed and thick. Cytokinesis is the division of the cytoplasm, the actual cell splitting into two. This takes the mother cell's cytoplasm and equally kind of separates everything up, divides everything up, and then encloses each daughter cell in its own plasma or cell membrane. It will often start during anaphase, so there is some overlap between cytokinesis and the stages, the PMAT, the stages of mitosis. It looks a little bit different in plant versus animal cells. Why is that? Well, plant cells have a cell wall, whereas animal cells do not. Okay, so in animal cells, what begins to happen is what we call a cleavage furrow. Okay, this is a contractile ring forms in the middle, and it just starts to pinch the two together, like taking somebody's belt and pulling it tighter till they pop into two. Okay, like pulling on a drawstring, and then this pinches the cell into two daughter cells. This slide is showing cytokinesis happening in an animal cell. And again, you can see this cleavage furrow here, this indentation, okay, in the contractile ring here in the center, pinching that cell apart. And as you go from this to this, you can see it slowly stretches and pulls until, okay, and it forms a new membrane around each daughter cell. We can do that because there's no cell wall, and the cell membrane is much more flexible, and you can just pinch it into two. In a plant cell, it's a little bit different because between the two cells, you have to make a new cell wall. That becomes with the formation of a cell plate. Okay, they do not permit that furrowing, that cleavage furrowing, or that pinching in. So many small membrane-bound vesicles made by the Golgi apparatus are sent to the middle of the cell where they fuse together to make one thin vesicle stretching right through the middle of the mother cell. These membranes of the cell plate then become the plasma membrane, and the space between them gets filled with middle lamellae, and then they later secrete the primary cell walls, which is usually cellulose, and that's kind of put on the outside of that so that you have cell membranes, middle lamellae, with a cell wall then dividing the two daughter cells from each other. Again, here you can just see a picture of this forming. Okay, out here's the plasma membrane of the mother cell. We're really zoomed in here, but you can see the cell plate starting to form here all the way across the middle of the cell. Okay, so vesicles here that have membrane, like a cell membrane, a phospholipid bilayer, and those will eventually fuse so that you have a membrane here and you have a membrane here. So you have a cell membrane for each of the daughter cells. Okay, last part of section 3 talks about the functions of mitosis. Okay, why would you need to make more cells? Well, for growing, getting bigger, and also for repair, to damage like a cut, in order to grow back together. On flowering plants, later in the spring, when we do our plant unit, we'll talk about meristematic tissue. Meristematic tissue retains this ability to go through mitosis and divide and form new tissues, more cells. It's how trees, as they grow, get wider in girth or width each growing season. In mammals, mitosis is necessary once, sperm egg makes a fertilized egg and you're an embryo. It has to divide and add more and more cells to become a fetus. And then, when you're born, a child, and then eventually an adult. So growing as you get older, okay, you get bigger. You're not the same size as when you were born. I hope not. Okay, cuts, broken bones, those kind of things need more cells. So you have to stimulate cells to go through mitosis to fix those, to grow or to repair. Tissues that have a very prolific way of going through mitosis we call stem cells. Many mammalian organs contain stem cells. They retain the ability to divide and make new tissues. For example, our red bone marrow, we talk about in Bio 2, makes all types of blood cells. Okay, all types of blood cells. And so what we're trying to use is use these stem cells, these cells that have the ability to become lots of other cells to divide and specialize into two types of cloning, therapeutic and reproductive. Therapeutic cloning is to produce a certain human tissue, a certain human organ. If someone with leukemia, which is a blood cancer, can you stimulate stem cells to form a new healthy bone marrow? And we can either use adult stem cells or embryonic stem cells, which are a little bit more controversial. Or a little bit later, we'll talk about induced pluripotent stem cells, or iPS cells, on which scientists have actually taken adult cells and kind of tricked them into acting like embryonic stem cells, so that there's a more potency for the types of tissues they can become. Reproductive cloning, we talk about like with Dolly the sheep, where you're taking an individual and trying to take stem cells and clone or make a whole new organism from that group of stem cells. Again, this slide is showing the difference between reproductive cloning, where you remove the nucleus from one cell, put it into the egg cell of another, try to culture it into an embryo, plant it into a surrogate mother, and get a whole new organism or clone, versus therapeutic cloning, where you're going to remove the nucleus, put the nucleus in the egg, and try to get those embryonic stem cells here, or adult stem cells, to grow into a certain type of tissue like nerves, blood, muscle, especially nervous tissue or muscle tissue, things that normally are in G0 and you won't heal themselves. Can you get stem cells to grow and divide and cause those types of injuries or malformities to function properly again? Okay. So, sorry I forgot to add this in the first time. So, this time it is panning out.